Hi, I'm Andrew Filer. So I'm a clinical academic here at the University of Birmingham and the Queen Elizabeth Hospital. So I lead Birmingham Tissue Analytics Facility in Birmingham. And this is a purpose-designed facility where we use high-dimensional platforms to analyse outcomes in tissue. So our work is both with our Birmingham academics and with industry partners, where we take tissues and we use either spatial proteomic or spatial transcriptomic platforms to analyse those tissues. And I guess a lot of our work is either in discovery science, in discovering mechanisms of disease, and increasingly it's also working with industry on clinical trial outcomes. So it's vitally important to us that we have robust platforms that are proximal to translation into the clinical zone. Yes, so we've got some really robust platforms already in place that make our facility work. What we miss in terms of our ability to do multiplex antibody staining is that higher plexity. So when we're looking for a new machine, we wanted a machine that would deliver robust higher plexity. So it needs to have a very good automated pipeline. We can't work with a platform where we're going to have to be removing cover slips, or where there's a microfluidic platform that's flaky and needs to be replaced all the time. We're happy that Comet delivers that. We need speed, and we need something that's very robust and repeatable. And actually, the Comet just occupies that beautiful Goldilocks zone where we have all of those things in place. And the bonus is we can be looking at 4 tissues at once on a run. So that's massively important for us. So for other facilities, Comet has massive potential benefits. So it's great for discovery science, obviously the high plexity, that robustness of the microfluidics platform, the high throughput. With our experience, certainly we've got multiple lower plexity platforms on existing machinery that we use, and we've been able to combine those relatively easily because we can put those almost directly into Comet when they're well-known antibodies we found they perform very well. Just do limited validation through the cycle, and actually you can combine them to produce these bigger than stock multiplex panels. So it works very robustly, so it's great for discovery work. But again, because of that robustness, because of the automation, it also means you can start to use it for more demanding applications. And so we're looking at using it for proximal to GCP work, for preclinical work as well, because we're getting robust and repeatable work. We have good use cases actually across clinical samples in cancer and COVID that have worked really nicely with that. So it's very attractive to the facility that's delivering across a broad. So Comet for us in our unit, and for other discovery scientists and also people working in industry, there are some key scientific applications of this, obviously, and the most pressing one at the moment is single cell work. So here in Birmingham we do a lot of single cell work. We're leaders actually in some areas, particularly in my area of rheumatoid arthritis. And clearly if you're going to start discovering 70 plus different cellular subpopulations in the synovium, you've got to have a high-plex platform to be able to do the validation for that. And Comet absolutely hits that mark. So this whole single cell revolution that we're trying to cope with at the moment, Comet is the ideal platform for that. We have enough plexity that we can use 2 or 3 markers for every different cell population in order to validate our findings and extract that key spatial information. We first heard about Lunaphore and the Comet system actually when it was brought to Birmingham to demonstrate, and we immediately recognized it as the type of platform that we were after, because we wanted something very high plex, something robust. And actually all of the other platforms that were just coming to market because this new technology, they all had one major failing. So the reason we like the Comet is that it doesn't have any major weakness. It manages to cover all bases, and that's really important to us here. So Comet integrates into our workflow very nicely. In one important respect, because we can use our own antibodies, we can transfer our lower plex platforms immediately into the higher plex ones, validate them quite quickly. So that means we're not starting all over again trying to buy new antibodies. That's really important for us. The outputs of Comet we can put directly into our other platforms. So there's already a collaboration with Visiopharm, which is an analysis platform that we have, so we can do multiplex analysis and clustering through the Visiopharm apps as well. And it also fits with our spatial transcriptomics platforms as well. So we can put all of those data, we can export the data out of the Comet and analyze those at the same time. We've got nice usage cases of that recently, particularly around COVID.